Aloha, and welcome to yet another lovely pre-recorded lecture. In today's lecture, as new material, we are going to cover chapter 12 of your textbook, which is all about different processes of cell division. But before we do so, let's start off with a little bit of review. And before we begin with that review, here is yet another lovely picture of Benedict Cumberbatch. All I have to say about this picture is wowza. Now let's review a little bit about Chapter 11. Chapter 11 is all about a process that just about every cell that's out there should be capable of doing, and that's something called cell signaling. Cell signaling describes a type of communication that occurs between cells, whether they be prokaryotic or eukaryotic. And without cell signaling, organisms wouldn't be capable of many of the characteristics, like the ones that you see here, that are part of this list. So without cell signaling, your body wouldn't be able to regulate itself. Without cell signaling, you wouldn't be able to respond properly to your environment. So whether prokaryotic or eukaryotic, all organisms should be capable of some form of cell signaling, and by doing so, many of the basic processes of life are capable of happening. So let's talk about how cell signaling itself happens. And really the answer to that question, how does cell signaling happen, depends upon how close together those communicating cells are to each other. So on the left again, if you recall from the last lecture, these are some examples of cell signaling occurring between cells that are physically touching, say through the help of gap junctions, if these are two animal cells that are part of a tissue, or maybe plasmodesmata, which are structures built between plant cells that are part of a tissue. So these are forms of cell signaling involving direct contact, and these cells are always in direct contact with each other. There's another type of direct contact through which cell signaling can occur, and it's with the help of the cell surface biomolecules, all those biomolecules that we may have talked about that makes a plasma membrane a mosaic, like different kinds of carbohydrates and different kinds of proteins. Many of those cell surface biomolecules have compatible shapes with each other, and that's what allows them to physically interact with each other, like is shown here, to allow cell signaling to occur. And this is often the mechanism that's employed by, say, cells that are part of your immune system that are meant to recognize all the other cells in your body as being part of you. Or if it's something that's not part of you, recognize that as being foreign and get rid of it eventually. So that's another kind of cell signaling that applies to cells in direct contact, but only temporarily. There is yet another mechanism of cell signaling that specifically applies to cells that are not in direct contact. And depending upon how much space is between these cells, slightly different ways of getting this done. So at the top of this diagram, two forms of cell signaling between cells that are separated by a small amount of space, and at the bottom, same kind of cell signaling that's occurring between cells that are separated by a large amount of space. So let's start off with the kind of cell signaling that occurs between cells that are separated by a small amount of space. In general, it can be called paracrine signaling, because like parallel lines, they can be separated by a small amount of space, be really close to each other, but they will never ever touch each other. And if we're talking about paracrine signaling as it applies specifically to neurons, we can name it after the small amount of space that separates a neuron from its intended target cell and call it instead its synaptic signaling. In both cases, the cell that's doing the communicating is called the signaling cell, like this neuron is acting as a signaling cell. It has to produce, this signaling cell, some sort of a substance. Usually it's a molecule, like a neurotransmitter, an amino acid-like molecule, or a steroid, or something larger like a protein. Those molecules, whatever they might be, produced by the signaling cell and then secreted through a process that you already know about, thanks to chapter 7 of your textbook, called exocytosis. So it's packaged up into vesicles. Those vesicles travel to the cell membrane, and that substance, usually some sort of a molecule, like a biomolecule, secreted out of the cell. It travels through diffusion, simple diffusion, across that small amount of space, and thus is received, reaches and is received by its intended target cells, which are nearby. If we're talking about this kind of signaling as it applies to cells that are separated by a large amount of space, we would call it, instead of paracrine or synaptic signaling, either endocrine or hormonal signaling. And that's the reason why many of these secreted molecules are called hormones, as sort of a general term. So it's the same mechanism, it just happens across a much larger distance. So here's my signaling cell. Sometimes it's called an endocrine cell, named after this type of signaling. It makes some sort of a molecule. It could be a protein or a steroid that acts as a hormone. And through the process of exocytosis, this hormone or signaling molecule is secreted out of the cell, but into one of two different bodily fluids, typically either the lymph or the lymphatic system, or into the blood or the bloodstream. And it's the lymphatic system or the bloodstream that carries these signaling molecules across a much longer distance so that they will eventually reach their intended target cell that is nowhere near the endocrine cell that was initiating the contact in the first place. So overall, the way in which cell signaling occurs thanks to one of two different basic mechanisms, depending upon how close together those cells are to each other. One mechanism applies to cells in direct contact. Another basic mechanism or two applies to cells that are separated by either a small amount or a large amount of space. So let's review the basic steps that are involved in cell signaling overall, regardless of the specific mechanism by which it happens. And just for the sake of simplicity and clarity for this review, like last time, we are going to focus on not the signaling cell, the one that initiates the communication. We're going to focus all of our energies on the cell that's supposed to be receiving the message, and more importantly, responding to it correctly, the so-called target cell. So that's what's shown in the textbook diagram. This is the small amount or large amount of space, which is labeled as the extracellular fluid, that separates this target cell from the signaling one that's either close by or far away. This right here is the cell membrane of that target cell. And in this diagram is an outline of the three different steps that are all the responsibilities of this target cell that's in question. So the first step, which makes a little bit of common sense, is like your friend that should have his cell phone on to get the message. And this first step is called the reception step because that's the intention of it, making sure that the target cell receives this message properly. All right. The next step for this target cell is, once received, that message must be transduced. So the second step is called signal transduction. And what I told you last time is the word transduce or transduction just means to relay or to pass something on. So here we are trying to pass that message on to some other part in the cytoplasm of this target cell that is supposed to create the proper response. So signal transduction is all about passing that message on and passing it along properly to some other part within the target cell. Last but not least is the whole reason why cell signaling in any form happens. It's to get this target cell to respond somehow. And in the last lecture, I told you, and also sort of demonstrated by being very vague, that this is the most vague part of my lecture because it can be just about anything, the response that the target cell is supposed to mount. So let's talk about these three basic steps in general, and then maybe as an illustration of all of them, I'm going to use Lance Armstrong and why he got in trouble, just to illustrate how these three steps work in, say, the human body. All right, so first step for this target cell is to get the message properly. And in order to do that, the target cell has to properly make and also put in the right location a molecule called a receptor. Now, we talked about in the last lecture these receptors being proteins, and therefore, like any other protein, it should have the proper three-dimensional shape. And that some of these receptors come in two basic forms. One receptor, which is called by your textbook a cell surface transmembrane receptor, is embedded in the cell membrane. So like this one, if it's a cell surface transmembrane receptor, like this one is, it's a protein that's stuck in the membrane. And what that really means is it's immobile. It can't leave the cell membrane. It has to stay there. There are other receptors that are floating around freely in the cytoplasm, which means that they are mobile and capable of moving around in the cytoplasm. These kinds of receptors, they're also proteins. They also have to be made properly in the right shape by these target cells. They're called because that's where they are, inside, intracellular receptors. So receptors come in one of two different forms depending upon where their physical location is. Cell surface transmembrane receptors are proteins bound to the cell membrane, whereas intracellular receptors are proteins that are mobile because they're free floating in the cytoplasm. So that's what the target cell has to provide in order to get this reception step done. What the signaling molecule provides that may have to travel some distance to get to the target cell is this. So this could be a steroid, it could be a neurotransmitter, it could be a protein. Whatever the signaling molecule is called in general ligand or ligand. And it's called that because its job is to ligate or to bind to a specific part of this receptor. So whatever is acting as a signaling molecule, sometimes we call it a ligand or ligand. And it's called that in general, these signaling molecules, they're called ligands because they bind to a specific part of a receptor, and that part is called the ligand binding site. So like the active site of an enzyme, the job of this ligand, because he's got a compatible three-dimensional shape to that ligand binding site, is to bind to temporarily the ligand binding site of this receptor. So again, just about anything that changes the overall shape of this receptor may also alter the overall three-dimensional shape of that ligand binding site, inhibiting this temporary binding from occurring. So that's basically what occurs as part of the so-called reception step of cell signaling in order for this target cell to quote-unquote receive the message properly. The second basic step is called the transduction step. And in the transduction step, that original message that was received by some sort of a receptor has to be passed on to some other part within the target cell. And this happens one of two different ways. It depends upon what kind of receptor is getting that message in the first place. Cell surface transmembrane receptors like this one that are bound to the cell membrane and can't go anywhere else by themselves, they rely upon in the signal transduction step a whole host of these relay molecules. These could be proteins. They could also be an organic molecule called cyclic AMP for short. Whatever these relay molecules are, they must be in the proper three-dimensional shapes themselves in order to get this message from the receptor and also pass it on to each other. And very often this involves the passing on of phosphate groups from one relay molecule to the next, like a little baton that's being passed in a relay race. The addition of these phosphate groups to the next relay molecule will get this message to where it's supposed to go. Intracellular receptors that are not bound to the cell membrane like this one is, intracellular receptors that are mobile and free-floating in the cytoplasm, they don't rely upon relay molecules of any kind. Because they're mobile, it's the receptors themselves that will transduce the message without any other outside assistance. So depending upon what kind of a receptor gets the message in the first place, that's how this process called signal transduction will happen next, whether it's a cell surface transmembrane receptor that gets the message or an intracellular receptor that gets the message. The mechanism of signal transduction will differ from each other. Last but not least, whichever way we've relayed that message to where it's supposed to go in the cytoplasm of this target cell, the last basic step for this target cell is the whole point of cell signaling. It's to create the correct response. To create the correct response that's appropriate to that particular message. So this response can be just about anything. Maybe you are stimulating the cell to reproduce through some form of cell division. Maybe we're trying to suppress or inhibit cell division. Maybe we're trying to activate an enzyme in the cytoplasm. Maybe we're trying to make cellular respiration happen faster or slower. Just about any function that this target cell is capable of is possibly responding to that message. And there are many different factors that determine what that proper response is supposed to be. Some examples could be what the target cell is. Another factor is what receptors it has. Another factor might be what the signaling molecule or ligand is. Sometimes it's the amount of the receptor that plays a role in what the response is going to be. Sometimes it's the relative concentration of the signaling molecule. Sometimes variables that are involved in what the response is, what relay molecules transduce that signal. So just about anything that a target cell does is possibly what the response is going to be. It can not only be an activation of that function or a turning off of that function, it can also be a dialing up of the volume, if you will, on that particular function, making it happen faster or more frequently. It can also be a dialing down on the volume, make it happen slower or less frequently. So again, response step, just about anything that a target cell is supposed to do, possible response to that message, and many factors determine what that proper response is supposed to be. So here's an example using Lance Armstrong of the three basic steps: the reception one, the signal transduction one, and finally the response one for a target cell in cell signaling. If you recall, a few years ago, Lance Armstrong got into a lot of trouble for something called blood doping. He was shooting his body up with many different hormones that are designed to enhance his athletic performance. And one particular one that he was accused of taking is something called EPO for short. And EPO stands for the full-on name of a protein called erythropoietin. And this protein is produced, at least in adult humans, in your kidneys. So EPO is a protein that's made by and secreted by adult human kidneys. And the response that EPO is supposed to create occurs in the bone marrow, in select cells in the bone marrow that are supposed to divide and eventually turn into red blood cells. So let's talk about one of these cells. So here is in the bone marrow, one of these special cells, and this will be the membrane of that special bone marrow cell. All right. On the surface of these bone marrow cells should be a special kind of cell surface transmembrane receptor. That is a transmembrane receptor. I'm going to embed it in the cell membrane. All right. And this is the ligand binding site of that particular receptor. We're just going to call it, for the sake of simplicity, an EPO receptor. And like any other receptor, This EPO receptor is a protein, and therefore he's got a very specific three-dimensional shape. So number one, in the reception step, what has to happen first is that this EPO has to be secreted by the kidneys through that process called exocytosis. And the bone marrow is very far from the kidneys very often. So in order for this EPO to get to where it's supposed to go in the bone marrow, it has to travel through the bloodstream. So EPO secreted by the kidneys. Next, it ends up in the bloodstream. And the bloodstream carries that EPO molecule through the bloodstream to get it into the bone marrow. And I'm just going to make it a little ball. That EPO eventually, because it's got a compatible shape to the ligand binding site of this EPO receptor, it will temporarily bind to this EPO molecule, temporarily bind to the ligand binding site of this EPO receptor and be held there using weak bonds. All right, so this is the reception step, getting that EPO to bind properly to the ligand binding site of this EPO receptor. The second basic step is, once this EPO receptor has a molecule of EPO bound to its ligand binding site, the transduction step, or signal transduction. Since this EPO receptor is stuck in this bone marrow cell's membrane, it can't go anywhere on its own, this receptor will temporarily change shape. And that temporary change in shape in this protein or EPO receptor will activate a whole host of other signal transduction molecules, relay molecules that will get this message to where it's supposed to go. So we're just going to say a whole bunch of relay molecules are activated next. So this activated EPO receptor will activate these relay molecules very often by adding phosphate groups to them to activate these relay molecules. Those relay molecules will probably go on and activate other relay molecules that activate other relay molecules, and on and on and on to disseminate that message to different parts within the special bone marrow cell. In the end, the third step, once we've relayed that message to many different parts within the special bone marrow cell, is the response step. And the response that should be triggered in this bone marrow cell is cell division. So basically, this cell will be signaled to divide into two, to reproduce, to make more of itself, and eventually those new cells that are created from him by him dividing into two, and so on in that process of cell division, causes the production of new red blood cells. So with extra red blood cells being produced in Lance Armstrong's body, all of which was triggered by this EPO hormone, he has greater oxygen carrying capacity and thus greater endurance when scaling the Alps as part of the Tour de France. So Lance Armstrong and the blood doping that he was committing, really an example of the three basic steps for a target cell in the process of cell signaling. Chapter 12 is all about how cells are capable of reproduction, and they would normally reproduce through some process of cell division. So cell division happens in basically all cell types, but for many different reasons. So let's cover some of the basic reasons why cell division might be important. If you're a unicellular organism, cell division might be your only means of reproduction. And this is especially true if all you're made up of is a single cell. So here's an example of an amoeba. And some of you saw amoebas when we did Lab 4 together many weeks ago. For amoebas to reproduce, basically one amoeba cell will divide into two. So the offspring, if you will, are those two new cells that are generated as a product of cell division. Sometimes, especially in a multicellular organism, cell division happens for different reasons, and therefore different kinds of cell division might be possible. Here are some examples of the reasons why in a multicellular organism cell division might be important. In order for us to reproduce, we have to do a type of reproduction called sexual reproduction. Now, in sexual reproduction, it's a little bit more of a complex process because in order to get it on with somebody else in sexual reproduction, each parent first has to create cells that are called gametes. And in humans, these gametes are either sperm or egg cells. The production of those sperm or egg cells that occurs in a human being has to do with a special kind of cell division called meiosis that occurs within that human being. So through one mechanism of cell division, we get to produce our own personal gametes. And once fertilization happens, that offspring that we make, which is a fusion between a sperm and an egg, has to undergo several rounds of cell division in order to grow and in order to develop different tissues in the process of embryonic development. So another kind of cell division takes place in order to allow for that growth or eventually that development of different cells into different tissues. In a fully fledged multicellular organism, like in a human being, when injured, there should be some kind of wound healing that's going on. Well, wound healing is basically your body making more of a specific tissue type. So for the purposes of tissue renewal, cell division could be happening as well. So really the bottom line of this slide is cell division is important. It's important for many different kinds of cells, and it's important for many different reasons. So let's talk about one really important thing that must happen before any form of cell division is capable of happening, and that's something called DNA replication. So we talked about DNA in any cell acting as a set of protein recipes. And if you're going to reproduce that cell, you have to pass on a set of these protein recipes. That's the reason why before cell division in any form happens, DNA replication has to really happen first. So this is something I just want you to file away for now. We're going to come back to the process of DNA replication later on in future chapters and talk about one step at a time how it actually works. But for now, keep in mind that for any cell to be truly ready to divide, regardless of what type of cell division it does, DNA replication is one really important preparation for any form of cell division. So let's begin by talking about cell division as it applies to prokaryotic cells. And lucky for you, this one slide is the only slide in my entire lecture that will describe how prokaryotic cells divide. And it's not mitosis. This process of cell division is not called meiosis either. Prokaryotic cells divide by their own special mechanism, and it goes by a completely different name. It's called binary fission. All right, so let's talk about this two-word term, binary fission, because what it really describes is literally what's happening to a prokaryotic cell in the process of it. So here's a pretend prokaryotic cell. It happens to be an E. coli bacterium, but this could also be an archaea as well that's undergoing this process, so prokaryotic cell of any kind. The term fission is the exact opposite of the word fusion. So what that describes is a splitting. And the word binary describes the end result of the cell splitting. In the process of binary fission, you form two offspring or daughter cells. So they are called daughter cells, the end result of binary fission. I'm not sure why they're not called son cells. For some reason, we are sticking with the feminine and not the masculine term, all right? So binary fission is the specific name applied to prokaryotic cell division, and this two-word term really describes what's happening in the process of it. Now let's take a slightly closer look at the process of binary fission as it applies to prokaryotic cells. One important thing that's shown in this diagram is DNA replication. So if you recall from many lectures ago, inside of a prokaryotic cell, there is no true nucleus, but there is DNA in there, and it's in a location called the nucleoid. So this purple tangle represents the DNA found in the nucleoid of this prokaryotic cell. And it's a single molecule of double-stranded DNA. It's also a circular molecule of double-stranded DNA. In the process of binary fission, DNA replication is also happening. So that's really the beginnings of what you are seeing here, that single circle of double-stranded DNA, at the same time the cell itself is dividing into two, the single circle of double-stranded DNA is getting copied or replicated. And as the cell divides, those two copies, this is one, the original one, of that circular DNA molecule. Here's the extra copy of that circular DNA molecule that's being produced. Those two copies of this circular DNA molecule are getting pulled apart in the process of binary fission. And the reason why they're getting pulled apart is they're anchored to opposite ends of this dividing cell, and the cell itself is getting longer and longer and longer. So what it's really producing more of is two things: cell wall, and that is one major thing that the antibiotic penicillin will shut down in a bacteria cell, prevents a bacteria cell, penicillin does, from producing more cell wall material, as well as more plasma membrane. Because if you want to get longer and longer and longer, you have to make more of these two things, both cell wall and also plasma membrane that's right underneath that cell wall. All right. With those two things being produced, extras of that stuff, the cell lengthens, and then finally it gets long enough that something comes along, it's usually proteins, that help to cut the cell off and divide it into two separate daughter cells. And notice that in each of these two daughter cells, they have their own copy of that single circular DNA molecule. And we're assuming that DNA replication is 100% faithful. So we're going to assume that these two molecules of circular DNA are identical to each other, which means that these two daughter cells should essentially be clones of each other. They should be completely identical, at least at the DNA level, completely identical to each other. We've just made extra copies of that parental or original cell. This happens pretty rapidly. In an E. coli bacterium, it takes roughly, in E. coli, about 20 minutes to go from one parental cell into two separate and identical daughter cells. Not only that, like I just told you, DNA replication is happening at the same time as binary fission. So in the same 20 minutes, it takes this E. coli to divide into two. It has also completely generated a new copy of its original circular molecule of DNA. All right, so that covers the gist of binary fission, the special kind of cell division reserved for prokaryotic cells. Eukaryotic cells are much more complex. One reason why is their internal structure is much more complex. They have a nucleus. They have other organelles that they have to divide up as well, like mitochondria, maybe chloroplasts, rough ER, smooth ER, Golgi apparatuses. All of these things also have to be divided up if that cell is going to divide into two. Because of its sheer more complex structure, the entire process of eukaryotic cell division is also more complex. Likewise, in the nucleus of a eukaryotic cell, there is much more DNA to take care of in the process of cell division. So let's focus on the DNA for now before we move on and talk about the kinds of cell division that are possible in eukaryotic cells. So at the top of this diagram, which unfortunately is not in your textbook, this is part of the DNA that's found in the nucleus of a eukaryotic cell. It isn't a single molecule. In a human nucleus, there are several, 46 total, very large molecules of double-stranded DNA. The DNA itself, not only is it double-stranded, it's not circular, it's linear. And like I told you many lectures ago, in order to help this sheer large amount of DNA fit into a very compact nucleus, we wind that DNA around these special protein spools. So these yellow blobs represent clumps of proteins that help that DNA wind and compact enough so that a whole lot of it, 2 meters worth of human DNA, fits into a single human nucleus. So typically, when people show you diagrams of the DNA in a eukaryotic cell's nucleus, they'll show you a structure that looks like this. And they'll say that this structure is built of not just DNA, but also the protein spools that DNA is wound around. And that's what this material named chromatin is describing, not just the DNA, but also the protein spools that DNA is wound around. And this chromatin is compacted further because the protein spools are sort of attracted to each other to compact this DNA even further. So the DNA wound around these protein spools loops more and more and more until finally it forms a structure that looks kind of like this. And people will tell you this structure is called a chromosome. All right, so what this term chromosome describes is not just DNA, but also the protein spools that DNA is wound around, and also that DNA is really compacted up into a structure that looks like this one. Here's what's confusing though, all right? Chromosomes, not just that they're DNA wound around these protein spools or made up of a material called chromatin, these DNA molecules or chromosomes have to be replicated before cell division can actually happen. So here's what's confusing. This is a chromosome. This little cartoon right here is one chromosome in a eukaryotic cell's nucleus before DNA replication. And if you take a look at this chromosome, it has a little bit of anatomy to it. All right, so I'm going to redraw that anatomy on the next slide just to give myself a little bit more room. So I'm going to redraw that chromosome as sort of like a bowling pin shaped structure. That is one chromosome, one molecule of double-stranded DNA wound around proteins and compacted up very tightly. All right, so that is a single chromosome. You'll notice that every single chromosome has sort of a constricted portion to it that's roughly in the middle. That region that's sort of constricted, that's sort of like the waist on a person, is called this chromosome centromere. So because the centromere, sorry for my crappy handwriting, is roughly in the middle along this chromosome's length, that's the reason why it's called the centromere. It's roughly in the center. Centromere comes from that term. All right. This is what a chromosome looks like before DNA replication. So this is one chromosome. Oops. Before DNA replication. All right. Here's what's confusing. This is what that chromosome looks like after DNA replication. So what's shown here is the extra copy of this original chromosome that has been made. So this guy right here that I'm going to use a blue arrow to point to, just to make it different. All right. This guy right here is the extra copy of this original chromosome. This is still one chromosome. That's one thing that makes this very confusing. This is still one chromosome, all right? But now it consists of 2 separate and identical copies that are stuck together. And they usually remain stuck together at least at their centromere, and they will stay stuck together for most of the process of cell division. Now a little bit more about these confusing terms. So far we've covered the fact that after DNA replication, this entire structure is still one chromosome, but now it consists of 2 separate copies. All right, those 2 separate copies are each called a chromatid. So this is one chromatid right here, and this is a second chromatid. To make things even more complicated, that these are identical copies to each other, we can call them sister chromatids. All right, so to lather, rinse, repeat, this is one chromosome, but it consists of two sister chromatids. And if this sounds confusing to you, it's confusing to some of my colleagues too. So make sure that you understand what this vocabulary means, because I'm going to use the word chromosome a lot throughout the next couple of lectures. I will also refer to chromatids or sister chromatids a few times. Make sure you understand what these terms are actually describing for you. All right, so just to make sure that we have this clear, again, going back to this textbook diagram, this is one chromosome. This is that same chromosome after DNA replication. But before DNA replication, there is just one chromatid. That's this one right here. After DNA replication, there are two sister chromatids, identical copies, still stuck together at a location called the centromere, that constricted portion along their length. So now that we've talked about a little bit more about the DNA content in a eukaryotic cell, and really that's one major thing we're following in the process of eukaryotic cell division, the DNA inside of that nucleus, we can go on and talk about the mechanisms of cell division overall. For a eukaryotic cell, there are two basic kinds of cell division, and they happen in different cells for completely different reasons. So I'm going to use as an example of a cell that's capable of these two kinds of cell division, cells that are part of the human or an animal's body. All right, so eukaryotic cell division, one basic kind of cell division that just about every cell in the human body is capable of doing is called mitosis. Now, the overall goal of mitosis is very similar to the overall goal of binary fission, and that's to produce daughter cells that are identical to each other and also identical to the parental or original cell that we started off with in the first place. So if all you're trying to make is more of the same cell type, mitosis is the kind of cell division that you're going to do to make more of that particular cell type. So just about every somatic cell, meaning body cell in the human body, like liver cells, other cells, skin cells, if they want to make more of themselves, if they're capable of cell division, mitosis is usually the only mechanism by which they can make more of themselves. In the human body, there is just one cell type that's capable of the other kind of eukaryotic cell division, and that's the kind of cell that produces gametes. So these are cells that divide to make more, for us, sperm and eggs. So that's a misconception that I want to clear up right now. The sperm and eggs you have come from a special cell that divides by meiosis to make them. It's not the sperm and eggs themselves that really divide to make more of themselves. They come from a special cell that divides meiosis to create them. And it's this process, meiosis, that these special cells making the gametes are capable of doing. The entire goal of the second kind of eukaryotic cell division is completely different from mitosis, because here we want our sperm and eggs, our gametes, to be different from each other. We want our sperm and eggs to differ not only at the DNA level from each other, but also from that original special cell that started meiosis to create them. So the overall goal of meiosis is not sameness, it's differences. And this makes sense at a survival level. Sexual reproduction is all about making offspring that are different from you. And by making your offspring different from you, hopefully in the wilderness, they can survive a little bit better than you can. So this variety stems from making those offspring different from you at the DNA level. Those DNA differences start off in the process of meiosis, in the creation of the sperm and eggs that go on later to fertilization to make those offspring. All right, so the overall goal of meiosis is not sameness. We want daughter cells that are definitely different from each other, at the DNA level, and this is in the end going to ensure differences in our individual offspring. So unless they're identical twins, every single one of the offspring you create with your mate should be different from the next offspring. Before we talk about the process of mitosis or meiosis, we have to talk about first all the different preparations that a eukaryotic cell will undergo to get ready for either of these two cell divisions. So that amount of time spent getting ready for either one of these two cell divisions is largely represented by a pie chart that looks like this. And this pie chart is something that's pretty commonly known in the biology world. It represents time, this entire pie chart, and it's called the cell cycle. All right, so let's talk about what this pie chart or cell cycle really represents. Again, it represents time. And just so you understand and can relate to this cell cycle a little bit better, imagine that the entire pie is roughly 24 hours worth of time. All right. What's shown here as a very small yellow sliver of the pie, I'm just going to change my pen so I can circle it in this diagram. What's shown here as this very small yellow sliver is the very relatively short amount of time a eukaryotic cell spends dividing. Notice that it's only a small sliver of the pie. The rest of the pie, which is about 23 out of those 24 hours, is labeled as interphase. What interphase represents is not the time a eukaryotic cell is spending in the process of cell division. Interphase actually represents the amount of time getting ready for cell division. So notice that interphase is much longer than the actual amount of time labeled as M phase that cell is physically dividing. And M phase, the M stands for here mitosis, but it could also really stand for meiosis as well in the cell cycle. All right, so that is one take-home message about this diagram. If the whole pie represents a certain period of time in a eukaryotic cell, the part that's labeled as interphase is the amount of time getting ready for some process of cell division, either mitosis or meiosis. To make your life more complicated, we can subdivide interphase into three smaller phases. The first one is called G1, and the letter G stands for the word gap. The second phase, the second part of interphase, is a span of time called S. S stands for the word synthesis. And the last phase of interphase is called G2 for short. And again, the letter G stands for the word gap. So if all of the pie chart or cell cycle is 24 hours, G1 and G2 alone would be roughly 5 to 6 hours. 5 to 6 hours. M on its own, I told you already, was about 1 hour out of this pie. S of interphase on its own is roughly 10 to 12 hours. All right, so a couple of take-home messages about interphase. If we subdivide it into these three smaller phases, again, the letter G stands for the word gap. But that doesn't mean that this is a gap of time where nothing is happening in that eukaryotic cell. In reality, during both G1 and G2 of interphase, there are a lot of events that are occurring during these two gap phases, like the cell is growing, it's pumping out a lot of new proteins that will assist in cell division. It may be taking apart organelles like the Golgi apparatus, the rough and the smooth endoplasmic reticulum. So many events are actually happening during these two gap phases. These are just some examples of those events, but there are many others besides the ones that I've spouted off. I want to focus your energy on specifically S of interphase. So what S describes is, again, the word synthesis. And what that is describing, the word synthesis, the synthesis of DNA. So S stands for the word synthesis. And what that really describes is DNA synthesis. What that really means is it's here during S of interphase that DNA replication is happening during the cell cycle. So a couple of things about S of interphase that I want you to make note of. Notice that first, This is well before cell division, not during, like in binary fission. DNA replication is way, way, way before cell division even happens, all right? So S of interphase is way over here. Cell division doesn't occur until somewhere over here in the cell cycle. So S of interphase, where DNA is replicated in a eukaryotic cell, that phase actually happens well before cell division. Secondly, it's really long because if the whole pie is 24 hours, this is about half of the pie right there. All right, so it's not fast, DNA replication isn't, like it is in a prokaryotic cell. So even though DNA replication happens before cell division in all cells, the timing of it is completely different in a eukaryotic one. Last but not least, before we leave the cell cycle, a couple more things. Most of the cells in the human body are in no part of this cell cycle. Instead, many somatic cells in the human body are in a completely separate phase that is outside of the cell cycle called G0. That's where the cell is just living its best life, and it's probably doing cellular respiration and many other things as basic functions to keep itself alive, but it's not getting ready to divide, nor is it actively dividing. Some somatic cells stay in G0 for their entire lifetime. Other somatic cells can remain in G0 for a certain amount of time and then get ready to divide and divide, then go back into G0 as needed. So G0 is sort of a holding pattern that's completely separate from this cell cycle entirely. And it's just a cell healthy, doing basic cell things, but not preparing for cell division of any kind, nor is it actively dividing. Last important thing that I want you to keep in mind about the cell cycle is all the events that happen in it, regardless of what part of the cell cycle we're talking about, and the timing of all of these critical events is controlled in the cell cycle entirely by proteins. And there are many different proteins that are involved in regulating and controlling the cell cycle, telling the cell it should be in G1, telling it to move on to S of interphase, telling it to move on to G2, and so on. All of these events and the timing of the cell cycle regulated by different kinds of proteins. Here are two important examples of proteins. They're just acting as examples of proteins that might be in charge of regulating the timing and the events in the cell cycle. That they're regulating and controlling the cell cycle, we call some of these proteins cyclins. And they work together with enzymes, and I know they're enzymes because this name ends in ACE, all right, called cyclin-dependent kinases. So the way in which this works, and if you take other biology classes, you'll learn way more about this than I would ever expect you to know for the purposes of this class. In order for a cell to be in G1, there's a particular cyclin teamed up with a particular cyclin-dependent kinase that dictates to the cell, you should be in G1. There's another cyclin and cyclin-dependent kinase that transitions the cell to be an S of interphase. Yet another cyclin and cyclin-dependent kinase team up together to tell the cell it should be in G2, and on and on and on. So bottom line is, all the events and the timing of all those events in the cell cycle regulated very strictly by a multitude of different proteins. Cyclins and cyclin-dependent kinases, just two small examples of a whole mass of different proteins involved in regulating all of these events. This is important because if a cell loses control of the cell cycle, it might be dividing when it shouldn't be dividing. That's basically what a cancer cell is. It's a cell that's part of your body that's dividing out of control. It's ignoring all the signals that are meant to tell it to stop dividing, and it's probably just dividing irregularly and anyway out of control. All right. So regulation of the cell cycle by these proteins is really critical for all the events in cell division to happen properly. And if a cell is not ready to divide, some of these proteins like cyclins and cyclin-dependent kinases can actually program that cell to commit suicide or apoptosis. So let's talk about mitosis, the first possible kind of eukaryotic cell division. That's really going to be the theme for the rest of today's lecture. All right. Mitosis basically describes in eukaryotic cells how a cell can divide to produce daughter cells that are identical to it. So the whole point of mitosis is to create daughter cells that are the same as each other and also the same as the parental or original cell that you started off with in the first place. One important thing we're going to follow in this parental cell in the process of mitosis is its DNA content. So in order to describe that DNA content in the nucleus of this parental eukaryotic cell, we have to employ some important terms that I'm going to use a lot as a part of this lecture. All right, starting off with the fact that most animals and plants on this planet are examples of diploid organisms. So we're going to talk about this term diploid and what it truly means about the DNA content of these organisms. Again, most animals and plants on planet Earth are examples of diploid organisms. Another way of rewriting this term diploid is 2N. All right, so let's talk about this term diploid and what it truly means about organisms that are diploid. And I'm going to come back to this slide in a second. Let me just forward us over to a blank one so I can talk a little bit more about this term diploid. So diploid, most plants and animals on this planet are examples of diploid organisms. The latter part of this term ploid, whenever you see it, describes this: sets of chromosomes. All right, so that's what ploid describes. The prefix di, if you think about it, all right, di means two. So if you are a diploid organism, every single one of your cells, more or less, contains in the nucleus a total of two sets of chromosomes. Now, remember that a chromosome is one of these doohickeys, really large DNA molecules that are complexed with proteins to form one of these thick structures, right? So that's a chromosome. This is also a chromosome after DNA replication. But chromosome, again, describing one of these very large DNA molecules that's inside of the nucleus of a eukaryotic cell. And you should have two sets of them, of these chromosomes, if you're a diploid organism. Why are you diploid and just about every other animal and plant that's out there an example of a diploid organism? Well, you're the product of sexual reproduction. So you got one set of chromosomes from your dad, all right, and his sperm. I know you don't want to think about dad's sperm right now, but we're gonna. Another set of chromosomes you got from your mom. And in humans, each of these parents contributes in that set of chromosomes a total of 23 chromosomes. And that's the reason why every single human being should have a total of 46 chromosomes per human nucleus. 23 originally came from father. The other 23, comprising one set of chromosomes, originally from mother. And that most plants and animals on this planet are the product of sexual reproduction. That's why most plants and animals on this planet are examples of diploid organisms. All right, so that's what that term diploid truly means. It describes that a cell or an organism contains a total of two sets of chromosomes. Now, there's another way of writing diploid, and that's this term 2N. Again, the number 2 describes the two sets of chromosomes in that cell or organism, but this little letter N tells you specifically how many chromosomes are in a set, and that differs wildly from diploid cell to diploid cell, diploid organism to diploid organism. So the little letter N represents the number of chromosomes per set. All right, so here's an example of what a number could be for the little letter N. Humans. Human N, you can say, is 23, because you get a set of 23 chromosomes from your mom, that's N. another set of 23 chromosomes, that's another N, from your dad. If you're a Drosophila fruit fly, N equals 4. So a Drosophila fruit fly gets 4 chromosomes from his mom and another 4 chromosomes from his dad. So a total of 8 chromosomes only per Drosophila fruit fly cell. So bottom line is, many organisms are diploid organisms. And if they're diploid, they contain per cell 2 sets of chromosomes. But the number of chromosomes that are present per set will differ wildly from diploid organism to diploid organism. All right, so that's the meaning of that term diploid. So going back a step to this previous slide, we're just going to make this discussion about mitosis as general as possible by following any diploid cell, regardless of what the number is that little letter N represents. We're just going to follow any diploid cell that contains a total of 2 sets of chromosomes. The end result of mitosis would be 2 daughter cells. This parental cell divides to produce a total of 2 daughter cells. Like I told you before, the goal is to make these 2 daughter cells be identical to each other and also to the parental cell that you started off with. So if I start off with a parental cell that's diploid, each of these 2 daughter cells should also be diploid. They should have the same number of chromosomes as what you start off with. They should have the same number of chromosomes as each other. So the whole goal of mitosis is sameness. All right, so keep that in mind at all times, no matter what happens next when talking about the process of mitosis. So before we talk about what occurs in the process of mitosis, I want to show you a little video. And the video that I'm going to show you right now is of a eukaryotic cell. It's also a diploid cell. So this cell right here, and you can't see the membrane of it very well, but the cell membrane is somewhere out here. All right. In here, you can kind of see the outline of it. That's the nucleus of this eukaryotic cell. And these raised structures that you see are the chromosomes inside of this nucleus. Now, this is not a human cell. I'm just going to erase things so you can see things a little bit better. This is actually a cell from a newt. Get my pencil back. All right. So this is a newt lung cell. And newts, if you know anything about them, they're a type of amphibian. They can live both on land and in the water. For newts, though, they're diploid organisms, so they are 2N, which means that this cell should contain a total of 2 sets of chromosomes, but N for a newt is 12. So really, in this nucleus, there should be a total of 2 sets of 12 chromosomes, so a total of 24. 2 times 12 is 24. So there should be a total of 24 chromosomes present in the nucleus of this newt lung cell. So what you're going to watch right now is mitosis in this newt lung cell sped up over time. And what I want you to notice that's happening in this newt lung cell is what the chromosomes are doing, what the nuclear envelope, that membrane enclosure around the nucleus is doing, and last but not least, what the cell is doing. So you're going to follow those 3 things, what these chromosomes are doing in that nucleus, eventually what the nucleus itself is doing, and then last but not least, what the cell itself is doing in the process of mitosis. So that video is the coolest thing ever, and I've watched it 5 million times, and I could probably watch it 5 million more times again. Couple of things you should have noticed as you were watching said video. First, you should have noticed that the chromosomes were moving around in this cell. All right. Second thing you should have noticed, if you look very closely, is that eventually the nuclear envelope, which is this covering that makes up the outside nucleus, that actually disappears. So eventually, after a certain period of time, these chromosomes are actually in the cytoplasm of the cell, no longer enclosed by the nucleus. And then finally, which is my favorite part of the video, you should have noticed that the chromosomes themselves divided up somehow, and that physical division of the chromosomes happens well before the cell itself divides into two. All right, so important things to keep in mind in the process of mitosis as we're going through what happens throughout this process. So let's follow one of these chromosomes out of the 24 in this new lung cell and see what it's doing, at least on its own, in the process of mitosis. So that's this one chromosome that's shown here. This is that one out of the 24 chromosomes in that new lung cell before DNA replication. So if you recall, DNA replication is before mitosis even begins. All right, so DNA replication has to happen to this chromosome first. So I'm just going to draw a line here just to remind you that chromosome duplication, meaning DNA replication, is before mitosis. And that happens specifically during interphase, or S of interphase. All right, so this is hours and hours and hours before mitosis that chromosomes get duplicated. Now, this is that same chromosome after DNA replication. All right, so that's what number 2 is describing. So this is an actual picture of a chromosome after DNA replication. The original copy of it is the chromatid on the left. The extra and presumably identical copy is still stuck to it. That's the one on the right that is still one chromosome, but now what you see are 2 chromatids, which can be called more specifically 2 sister chromatids because we're assuming they're identical to each other. It's really only number 3 in this diagram that is what happens to this chromosome in the process of mitosis. Before the cell divides into 2, so this is one daughter cell, and that's a second daughter cell, what we are separating chromosome-wise are these sister chromatids from each other. So one sister chromatid ends up in one cell, the other sister chromatid ends up in a second cell. So there's some kind of a movement of these chromatids that happens to separate them from each other. And to make things more confusing, once they separate from each other, these sister chromatids are no longer called that, sister chromatids. Now they are each called a chromosome. So this is one chromosome in this daughter cell. This is another chromosome in that second daughter cell. What creates this movement to get this physical separation to happen between these sister chromatids is a structure made entirely out of proteins, and it's something called a spindle. That's what all of this yellow stuff in this textbook diagram describes. It's something called the spindle, and this structure called the spindle forms in that eukaryotic cell in the process of mitosis, or later on also in the process of meiosis, to not only move the chromosomes around, but also to help the cell in a little way physically separate into 2 daughter cells. So let's talk about the makeup of one of these spindles. And what it's largely made up of is these protein fibers you've encountered before called microtubules. That's what all of these yellow stringy things describe in this textbook diagram. And these microtubules have different lengths. They also have different functions as being part of the spindle. All microtubules in the spindle grow out of these little structures that are called centrosomes. All right, so in a spindle, there should be a total of 2 centrosomes. And eventually they're arranged so they end up on opposite sides of each other in that cell. And like hair growing out of a hair follicle, microtubules of different lengths will extend out of these two centrosomes in different directions. All right, so let's talk about the different kinds of microtubules that can be found and what their functions would be as part of this spindle structure. Sometimes it's called a spindle apparatus. These short microtubules, oops, where's my pencil? Right here. All right. These short microtubules growing out of this centrosome are part of a structure called an aster. So you can call aster microtubules. Aster microtubules, they're short, kind of like an asterisk or a little star sort of a shape. They extend out, but they're very short, these microtubules that make up that aster. The job of these asters is to anchor the spindle to different locations. So this end of the spindle will be anchored to this side of the cell, thanks to these aster microtubules. Thanks to the aster microtubules on the opposite side of the spindle, this side of the spindle will be anchored to the opposite side of the cell. All right, so that's the job of these shorter aster microtubules. You also have long microtubules growing out of either centrosome, and some of them are called by your textbook non-kinetochore microtubules. That's a really long term to say 5 times fast. The job of these longer non-kinetochore microtubules, if you'll notice, they're overlapping each other and they're not touching the chromosomes at all, which are these structures in that cell, not coming in contact with the chromosomes at all. The job of these non-kinetochore microtubules is to help the cell stretch and lengthen in the process of cell division. The job instead of the kinetochore microtubules is to move the chromosomes around, and also to help the sister chromatids separate from each other in the process of mitosis. So if I draw in my own crappy way, one of these chromosomes, there are a total of 6 of them in this pretend eukaryotic cell. 1, 2, 3, 4, 5, 6. There are 6 different purple structures. Each one is a chromosome in this eukaryotic cell. At the centromere, on either sister chromatid, there is a blob of proteins, sort of a black-looking blob of proteins. These blobs of proteins are called kinetochores. Now let's see if I can fit that in there. Kinetochore. All right, so kinetochores are complexes of proteins that assemble themselves at these centromeres, that constricted portion of a chromosome. And it's the job of those kinetochore microtubules from either side of the spindle. So here's some kinetochore microtubules from one side of the spindle, kinetochore microtubules from the opposite side of the spindle. They physically attach themselves to either one of these kinetochores. And because they attach themselves to kinetochores, that's why these microtubules are called, hey, kinetochore microtubules. So it's the job of the kinetochore microtubules to make contact with the chromosomes in this way, to move the chromosomes around, and eventually help this sister chromatid move in one direction and the other sister chromatid that it's stuck to in the opposite direction. So a word of warning to you, we're going to follow 3 important things in the entire process of mitosis. What the chromosomes are doing, what the nucleus is doing, and what this entire structure called the spindle is doing. So let's talk about the process of mitosis, one small phase at a time. And we're going to divide it up into phases just so there's a little bit more clarity. And you're probably more familiar with these phases than I would ever expect you to know for the purposes of this class. So yet again, in each of these phases of mitosis, we're really only going to follow 3 things: what the chromosomes are doing, what the nucleus in this eukaryotic cell is doing, and last but not least, what that structure that's largely built of microtubules called the spindle is also doing. And to make your life a little bit simpler, not only are we just going to follow those 3 things in a eukaryotic cell, we are going to oversimplify this eukaryotic cell. So in place of it having so many chromosomes like a human cell would, we're going to limit ourselves to a diploid cell. So it has a total of 2 sets of chromosomes, But instead of 46 chromosomes, like you would find in a human cell's nucleus, we're just going to limit ourselves to just 6 chromosomes. All right. And keep in mind that DNA replication has already happened before this entire diagram. So each chromosome has already been copied, and each chromosome consists then already of 2 sister chromatids. All right, so what's shown in this diagram first is not mitosis yet. So let me just change the color of my pencil. This is the tail end, this first panel, of interphase, which is that long span of time spent getting ready for cell division. Specifically, we are at the tail end of interphase because we're in G2 of it. So if you need to, I would draw myself a line right here just to remind myself what happens before the line is not mitosis yet. It's everything, some of the things that happens before mitosis even begins. All right, so 3 things. First thing you should notice is the 6 chromosomes are hard to see because the DNA that makes them up very relaxed, and that is the normal state of chromosomes when in a eukaryotic cell. They're not very highly condensed, at least not until the process of cell division begins. The nucleus, in the meantime, is completely intact, so that is the nuclear envelope. All of this is cytoplasm, all right, that sort of light brown space inside of the cell, and this is the cell membrane that covers the cell. The last thing is, and even though this isn't mitosis yet, it's already begun to form, the 2 centrosomes that microtubules will eventually grow out of, they start off as together in the cytoplasm, and they start to migrate in opposite directions away from each other. This is still before mitosis begins, that the spindle in this way has already begun to form. All right, so chromosomes are relatively relaxed during interphase. It's not mitosis yet. Nucleus is still completely intact, and the 2 centrosomes not on opposite sides of each other yet. They have to migrate away from each other. In the meantime, microtubules are growing out of them. So this gets us to the first phase of mitosis, which is called prophase. And in prophase, you'll notice the chromosomes themselves. Now you can count that there are 6 of them. 1, 2, 3, 4, 5, 6, because the DNA itself has condensed. What that means is that sheer amount of DNA has crunched up really tightly to make each of these chromosomes more physically compact. The more compact they are, the easier they will be later on to move around. All right, so the chromosomes condense during prophase of mitosis. The nucleus does nothing because it's still completely intact. And in the meantime, the spindle is still forming. So the 2 centrosomes, they started migrating away from each other before mitosis. They're still migrating away from each other to end up on opposite sides of the cell. And microtubules of different lengths, you'll notice here, are growing out of them. Aster ones, kinetochore ones, non-kinetochore ones. This gets us to the next phase of mitosis called prometaphase. In prometaphase, the chromosomes are condensing even further, so notice that they're even shorter than what you saw in the previous diagram for prophase. While they're doing that, the nucleus itself is dismantling. So that's something you saw in the video of the newt lung cell, that the chromosomes eventually end up in the cytoplasm. They're not enclosed in a true nucleus anymore. The centrosomes in the spindle, now they're anchored to opposite sides of the cell, and microtubules are growing out of them. And some of them have started attaching themselves, like these guys right here, to the kinetochores built into the sides of the chromosome. So those are the 3 main things that are happening that are part of prometaphase. This gets us to the midpoint of mitosis, which is called metaphase. And in this phase called metaphase, the nucleus is still gone, the chromosomes are still condensed, and the kinetochore microtubules that are attached to them, like these guys right here, they will push them to the midpoint of the cell. So it's a little push and pull between these kinetochore microtubules to arrange these chromosomes in the midpoint of the cell. This midpoint is something called the metaphase plate, but it's an imaginary plane, which is why there's a dotted line along this metaphase plate. It's just the plane along which these chromosomes line up like soldiers in formation in the midpoint of the cell. So the metaphase plate here is horizontal, but it could just as easily be diagonal or vertical, the metaphase plate. All right, so that is the main thing that the chromosomes are doing during metaphase of mitosis. With the nucleus being gone, they line up in the cytoplasm in sort of a single file line, the so-called metaphase plate. And it's the kinetochore microtubules in the spindle that are responsible for pushing them so that they line up along the metaphase plate. Anaphase, the nucleus is still gone. The sister chromatids are being pulled in opposite directions. So this sister chromatid is moving in one direction, and this chromatid is moving in the opposite direction. And now that they're separate from each other, that's called the chromosome, not a sister chromatid anymore. This sister chromatid is no longer called a chromatid anymore. That is also called a chromosome. So we are moving chromosomes in opposite directions from each other. And that happens to each of these chromosomes that have lined up in the midpoint of the cell. Their sister chromatids get separated from each other. The nucleus is gone, and it's the spindle microtubules that are responsible for moving these chromosomes in opposite directions away from each other. The last phase of mitosis is called telophase. Telo literally means end, and that is distinct from cytokinesis, which describes the cell physically dividing into two. That's the very last thing, cytokinesis, that occurs in mitosis. So before we get to that, let's start off with what occurs in the last phase of mitosis, called telophase. The chromosomes, once they've moved in opposite directions from each other, they will relax. So notice that they're much longer than they were in the anaphase diagram. That's because the DNA physically relaxes or unloops a little bit. In the meantime, the microtubules that were attached to them are no longer attached to them, and they fall apart. So notice that you don't see really any microtubules in the cell anymore because they dismantle in the process of telophase. Last but not least, the nucleus reforms, and now we have two nuclei, because one of them is reforming around these around these 6 chromosomes. The other nucleus is reforming around those 6 chromosomes. So this will eventually be the nucleus of one daughter cell, and this is eventually the nucleus of a second daughter cell. All right. So in telophase of mitosis, the chromosomes relax, the nucleus reforms, and the microtubules in the spindle dismantle. The centrosomes are still there. They're not going to go away, but the microtubules that were extending out of them do go away during telophase. And then finally, the process of cytokinesis describes this, the physical division of the cell, presumably equally, into 2 daughter cells. So at the end of mitosis, what we're assuming is these daughter cells are identical to each other. If I started off with a single parental cell that was diploid, each of these daughter cells is also diploid or containing 2 sets of chromosomes. I start off with a single parental cell with a total of 6 chromosomes in it. Each of these 2 daughter cells contains a total of 6 chromosomes in that nucleus. And these chromosomes should be identical to each other from one daughter cell to the other one. So to wrap up the process of mitosis, just a couple of more details about this, what happens during anaphase, and this, what happens in the process of cytokinesis. So let's start off with anaphase. And if you recall in anaphase, what's really happening is that these sister chromatids are being moved in opposite directions away from each other. So if I can call that slide back up again, all right? So one sister chromatid moved in one direction, the other sister chromatid in a chromosome moved in the opposite direction. So one of those sister chromatids is represented here. That's a sister chromatid. This blue blob is a kinetochore, that complex of proteins. And within that kinetochore are these proteins called motor proteins. All right. These motor proteins in a kinetochore are important because it's these motor proteins with the help of ATP that will walk this chromatid along, no longer called a chromatid, called a chromosome, walk it along the length of this kinetochore microtubule in this direction. So with the help of ATP, this movement of this chromosome happens in this particular direction. And it's a physical walking of this chromosome along the length of the microtubule that it's physically attached to. So that's how we get the sister chromatids to separate from each other. They are each individually being walked along these so-called kinetochore microtubules with the help of these motor proteins. As that walking along these microtubules happens, the microtubules themselves get shorter and shorter and shorter. So these tubulin proteins dismantle from each other to allow those microtubules to get shorter and shorter and shorter. All right, so just a little bit more detail about what occurs to separate chromosomes in anaphase of mitosis. Now, cytokinesis, as is shown in this slide, slightly different in a plant cell as opposed to an animal cell. And if you think back to the structures of these two cells, that difference has to do with the fact that a plant cell has a cell wall, whereas an animal cell is only covered by a plasma membrane. So let's start off with cytokinesis in an animal cell, that it's only covered by a flexible plasma membrane. All we have to do is pinch off that membrane. So that's what's shown here, all right? That pinching occurs because there's a little band of proteins that forms along this plane that we're going to pinch off that plasma membrane. It's a band of proteins that forms called a contractile ring. And this ring is actually a ring of microfilaments. And these microfilaments assemble together along the midpoint of that cell. And like a drawstring on a pair of pants, this contractile ring contracts. It gets smaller and smaller and smaller in diameter. And as it contracts, it pinches off that very fluid or very flexible plasma membrane to form, in the end, two separate daughter cells. This pinching off can occur even though plant cells have a plasma membrane. You can't really pinch off that plant cell because outside of that plasma membrane is that pretty rigid cell wall. So in place of pinching off a plant cell, what a plant cell will do instead in cytokinesis is it's going to build a wall in the midpoint of it. And this wall is built because there are little vesicles, and those vesicles travel to the midpoint of this plant cell. They deliver cellulose and other materials required to build that new wall. The vesicles fuse together to form new plasma membrane on either side, and in between there's layers of plasma membrane, all that cellulose and the materials that they're delivering woven together to build a cell wall. All right, so the take-home message of this slide is cytokinesis in an animal cell happens in a different way from a plant cell because animal cells don't have a cell wall, whereas plant cells do. This next slide is just a cool one. It shows you at the very tip of the root of an onion bulb, there's a lot of mitosis going on. All right, so here's a little snapshot of some of those onion root tip cells. Even though many of these cells are undergoing mitosis, they're not in the same phase of mitosis, which is what's really cool about this picture. The chromosomes are stained in dark blue, so you can see that here. And the cell wall is the yellow outline on the outside of each of these cells. And by chromosome arrangement alone, I can probably assume that this cell, this one I'll use black just to highlight it, this cell right here is probably in anaphase, just based upon the arrangement of the chromosomes themselves. Whereas this cell right here because the chromosomes are bunched up together, probably in metaphase of mitosis. So it's just a really cool picture, showing you that even though these cells are undergoing mitosis at the same time, they're not all necessarily synchronized or in the same phase of mitosis all at the same time. Okie doke. So a couple of last things to close out this lecture. One has to do with when a cell loses control of cell division. That's really what a cancer cell is. It's a cell in your body that has lost control of cell division. So normally somatic cells in the human body, they're said to be anchorage dependent, meaning in order to divide, these normal cells have to be physically attached to a solid surface. Likewise, normal somatic cells will only divide enough to fill the gaps up in between them. So they are said to be also, besides anchorage dependent, they are also density dependent, meaning that because they only divide enough to fill up the empty spaces between them, you should only be forming a single layer of cells along that solid surface. And once they come in contact with each other, they signal each other to stop dividing. So they actually inhibit each other from dividing any further, only enough to fill up those gaps. All right. What makes somatic cells different from their cancer counterparts is this. Cancer cells are anchorage independent. So they don't have to be fixed to any kind of a solid surface in order to divide. So even if they're floating around in growth medium, cancer cells are still capable of dividing. So even though they're not attached to a solid surface, they can pile on top of each other and keep on dividing and reproducing anyway. While they're anchorage independent, cancer cells also differ from their normal counterparts because they're also density independent, meaning that they will not only divide to fill up the gaps between themselves, they will just keep on dividing. So they will pile one layer of cells on top of the other and just keep on doing that because, again, they're not going to stop themselves from dividing once they filled up the empty spaces in between each other. All right, so in every single way, cancer cells are cells that are abnormally dividing, and they're dividing so abnormally, their characteristics in terms of cell division make them very different from their normal somatic counterparts. Now, anytime anyone talks about cancer, they will probably talk about tumors. And what a tumor is a mass abnormally dividing cells. And you may have heard of, when talking about tumors, that they come in different grades. And you can thus grade the severity of that cancer according to the characteristics of the cells in that tumor. So here's an example of a breast cancer tumor. All right, so these are breast cells that are abnormally dividing from a single abnormally dividing cell, forms a mass called a tumor. If that mass contains cells that stay localized, meaning that they don't invade neighboring tissues, you can call that tumor a benign tumor. So benign tumors are masses of abnormally dividing cells that stay in one relative location. They don't spread, they don't invade neighboring tissues, and they definitely don't spread to other parts of the body. Therefore, benign tumors are very often pretty easy to remove because the cells in them remain localized and do not invade neighboring tissues. All right. What makes a cancer truly cancerous is when it becomes malignant. A malignant tumor contains abnormally dividing cells that have the capability of invading neighboring tissues. So here's an example of a malignant tumor that isn't staying localized. Those cells can spread and start to grow and divide in neighboring tissues. Usually late stage cancers and some of the most hardest to get rid of are ones that are metastatic. Metastasis describes the ability of cancer cells to escape the tissues they started off in, and they do so by hitching a ride in either the bloodstream or the lymphatic system that carries them to remote locations elsewhere in the body, and they can start to set up shop and divide and grow just as comfortably there as they did in the tissue that they came from. So metastatic tumors contain cells that are not only invasive, they can also spread to remote parts of the body and start to create tumors there as well. So breast cancer tumors, very often the cells in them, if metastatic, they will spread to the liver, sometimes the bone and the bone marrow, sometimes to other tissues in the body like the brain or the kidneys. And these are breast cells that are landing in these other tissues and growing and dividing somehow just as comfortably as they were in the breast tissue. All right, so bottom line is cancer cells are cells that are dividing abnormally and very often out of control. And when you have a mass of cancerous cells, that mass is called a tumor. So that's the end of chapter 12 of your textbook. Now, what we're going to talk about in the next lecture is chapter 13 in your textbook, and that's all about the other kind of cell division, which is called meiosis. And meiosis happens for completely different reasons. All right. And it's the other kind of eukaryotic cell division, and it's done in the human body to create not sameness, but DNA variety. So we'll pick up in the next lecture. We're talking about meiosis as part of chapter 13.